And thank you for joining us today for this very exciting panel discussion to celebrate World Immunization Week, titled Game Changers, Vaccine Innovations on the Horizon. My name is William Moss, and I'm the Executive Director of the International Vaccine Access Center, where our vision is for a world free from adverse health, economic, and social consequences of vaccine-preventable diseases. IVAC is a center within the Johns Hopkins Bloomberg School of Public Health that was founded in 2009 by Oren Levine and subsequently led by Kate O'Brien, who's currently the Director of Immunizations, Vaccines, and Biologicals at the World Health Organization. This year, World Immunization Week comes at a critical time for global immunization. As many of you know, during the COVID-19 pandemic, we saw immunization levels decrease in more than 100 countries, with millions of children missing out on life-saving protection of vaccines. The theme for World Immunization this year is the big catch-up, and it's critical that we restore health services and immunization services to ensure that all people have access to immunizations they need across the life course to live long, healthy lives. The pandemic also led to many innovations in vaccines, both in terms of technology and in terms of the way we evaluate them, and certainly the speed in getting vaccines from early development into programs. Today, we will be looking towards the future of vaccines, a future where we can protect millions of more lives from some deadly, debilitating diseases, including malaria, tuberculosis, respiratory syncytial virus, and Shigella. Today, I'm honored to moderate a panel of outstanding international experts working on the cutting edge of vaccines and public health. Our first speaker will be Dr. Brigitte Gersling of the World Health Organization. Dr. Gersling leads the vaccine prioritization and platform work within the product and delivery research unit of the World Health Organization. Today, she will be discussing an exciting technology that has the potential to revolutionize the accessibility and delivery of vaccines, that is, microarray patches. Our second speaker will be Sir Adrian Hill, who's the director of the Jenner Institute at the University of Oxford. He is a leading researcher in immunology, vaccinology, and genetic susceptibility of infectious diseases, and he will be speaking about his exciting new work on malaria vaccines. Our third speaker will be my colleague, Professor Ruth Karen, here at the Bloomberg School of Public Health. Dr. Karen is a professor in the Department of International Health and the former director of the Center for Immunization Research and the founding director of the Johns Hopkins Vaccine Initiative. Today, Dr. Karen will be speaking about her work over the past several decades on vaccines for respiratory syncytial virus. You can read about her recently published, editorial in the New England Journal of Medicine, in which Dr. Karen outlines the hope and future of RSV vaccines. Our fourth speaker will be another colleague here at the Johns Hopkins Bloomberg School of Public Health, Dr. Kausar Talat, who leads a number of different clinical trials for a range of pathogens. And Dr. Talat will be speaking about advancements in Shigella vaccines. Our final speaker will be a close colleague of mine here at the International Vaccine Access Center, Dr. Rupali Lemay, who's the Deputy Director of IVAC. And she's involved in a very exciting new project funded by USAID on control and prevention of tuberculosis. And Dr. Lemay will be talking about TB vaccines. So we're gonna get started, and I'm gonna turn it over to Dr. Gersling to talk about microarray patches. Ita.
Thank you very much. Good afternoon, good morning, everybody. It's very nice to be here and part of this panel. Thank you so much for the opportunity to present on microarray patches, also known as MAPS. This is an exciting time for this novel delivery innovation. I'll touch on some of the candidates that are in the pipeline and also forthcoming data to look out for. But first, I'm going to start with a bit of background to the technology itself and the programmatic need for this novel delivery tool. So as everybody in this webinar knows well, we need to have an effective vaccine and an effective delivery strategy in order to have efficient and effective immunization. And the important part here is in terms of equitable coverage. The equity aspect of the delivery strategy is really essential. We need strategies that enable the vaccine to be accessible to hard to reach and marginalized populations, because in many cases, these are populations where there is very poor, restricted access to healthcare. In low-income settings, we can be seeing a situation where more than 50% of the cost to immunize a child is actually due to the delivery cost. And we know that it will cost more to reach the children in the last mile. So we need novel vaccine delivery technologies that can ease delivery and reduce the cost to healthcare program by reaching people that we can't currently vaccinate. Next slide, please. And the COVID pandemic has had an enormous detrimental effect on routine vaccination programs, particularly in infants. So while we've been able to vaccinate 5.5 billion adults, this is in many settings using the same healthcare workers that would otherwise be vaccinating children. And so we're, as Bill mentioned at the start of this webinar, we're now in a situation where we have unprecedented numbers of children who have not been vaccinated. And in 2021, 50 million children did not get DTP1. When we look at measles, the first dose of measles has dropped from 86% to 81%. And these are low levels that haven't been seen since 2007. Next slide, please. The result of the low measles coverage has been large and disruptive measles outbreaks with the highest rates in sub-Saharan Africa, really highlighting the risk that large immunity gaps pose. And responding to measles case and measles outbreaks continues to be a priority. Next slide, please. In addition, we have now many vaccines to deliver across the life course and a number of new vaccines that are coming through the pipeline. In the top row of this figure here, you see the vaccines that are currently recommended by WHO, either for global use or in specific regions. And at the bottom, you see the vaccines that are coming through in clinical development and are anywhere between 3 and 10 years away from actually being deployed. So we have several vaccines soon to come, and especially for infants, increasing pressure on the delivery strategies and the need to find new mechanisms and new opportunities for immunization. Next slide, please. So microarray patches are a vaccine product innovation that, if available, could significantly ease delivery. I'm showing you a picture of 2 microarray patch formats that are in clinical development for flu, measles and rubella, and some other vaccines that I'll come to in a moment. But the important thing here is that these are innovations that remove the need for vaccine reconstitution. They offer the potential for increased thermostability because they are dry vaccines. They offer the potential for dose sparing because of the route of administration, and I'll come to that in a moment. And importantly, they don't require a trained clinician for administration, offering the opportunity for community health worker administration. A single dose presentation rather than a multi-dose vial offers the opportunity to reduce wastage, but also removes hesitancy to open multi-dose vials and immunize kids who might otherwise miss out. The reduced weight and the need for, the lack of a need for ancillary supplies such as needles and syringes is another advantage. And the fact that it removes needles altogether helps with the end user acceptability and the perception of this vaccine being less painful. So there are a number of different advantages. Next slide, please. And I'm showing you here how the patch is actually administered on the arm. It's obviously a needle-free technology that is presented on a patch that is a coin size with or without an applicator that administers the vaccine through an array of hundreds or thousands of microprojections on the patch into the epidermis of the skin, and when applied, presents the antigen to a rich array of antigen presenting cells just below the surface. And this is why there have now been some data that demonstrates that administration with microarray patches does offer the opportunity for dose sparing, increasing the immune response in comparison to vaccination with intramuscular routes of administration. Next slide, please. So clearly there are a number of advantages and a lot of opportunity. And we had in 2008, the idea to come together as a consortium with Gavi, WHO, Bill and Melinda Gates Foundation, PATH, and UNICEF to really look across the range of vaccine product innovations that were at that time in development or at least conceptualized, and develop a framework to evaluate those prioritizations and identify priorities. So that work was done in 2018, and the consortium is called the Vaccine Innovation Prioritization Strategy. And we looked at 24 vaccine product innovations. And through the framework that we developed that was both quantitative and qualitative, microarray patches were identified as the most important vaccine product innovation. And that's where a lot of our work has been focused to date. Next slide, please. I don't have time today to go through all of the work that VIPS has been doing to support the vaccine map development. Just suffice to say that we have been assessing the optimal priority vaccines that would likely benefit from presentation on a microarray patch and assessing their value. This is because a key assumption is that vaccine maps will likely be more expensive than their existing presentations to procure, but that there will be lower delivery costs, particularly if we can increase the thermostability of vaccines, reduce the size, and ease the administration. And also because of the public health and socioeconomic impact of vaccinating hard to reach populations that we couldn't previously reach. Next slide, please. So here's an overview of the vaccine pipeline. And you can see that there are a number of influenza vaccines in the pipeline, as well as a COVID vaccine, hepatitis B, and Japanese encephalitis. But the candidates that we're really interested in as WHO and as VIPS are the measles and rubella microarray patches. And I'm very pleased to say that there will be a presentation at the PATH Microneedles Conference next month on the emerging phase 2B data from children in the Gambia. So please do look out for that announcement. But as vaccines progress to late stage development in phase 3, this becomes complicated because we really need to encourage the commercial partnership now between vaccine manufacturers and the map developer. And the question of the investment case becomes really critical. In the case of MR maps, there has been an investment case that has been developed by UNICEF that will soon be published. But the other key drivers for investment, of course, is understanding the demand and if and how the vaccine would be used if it were available. Next slide, please. And to this end, we've been leveraging the WHO Capacity Innovation Framework to engage with country stakeholders to address critical research questions. Please press again. Such as, would countries want to use MR maps? And if so, how would they use them? Would they be used in certain delivery contexts? What are the important attributes associated with those use cases? And also starting to explore what are some of the key decision-making factors that countries would look at as they make decisions about whether to use microarray patches versus the existing interventions, particularly if they do cost more to procure. And this capacity innovation framework is an extremely useful platform for us to begin to have discussions about innovations in the context of countries' vaccine immunization programs to identify what are really critical characteristics to inform TPPs, to inform potential demand forecasts for manufacturers, and for us to support dialogue with vaccine manufacturers and map developers. Excuse me, next slide, please. So, as I mentioned, importantly, the capacity workshops are conducted in the country and in the context of the immunizations systems barriers and strengths to identify really what could be important for MAPS. So it's an opportunity to validate the potential use cases for microarray patches, to find the critical product attributes, and to identify what are the important factors for decision making. Next slide, please. Excuse me. So we have conducted to date 2 workshops in Ethiopia and Indonesia at the end of last year. And you can see the participants that were involved in those workshops. And a number of them were really focused on policy making at the country level, but also looking at the perceptions of donors, particularly as we think about how to drive these products forward into the next stage of product development. There will be another workshop that is conducted in Guyana next week with 5 countries from the Caribbean region. And we will also have one in Indonesia, in Uganda in July. Next slide, please. Excuse me. I'm just showing you here some of the outcomes from the Ethiopian and Indonesian workshops. And you can see the Indonesian workshops in green and the Ethiopian workshops in brown. And the most important attributes that we took away from this workshop in the context of those countries' immunization system barriers was that it was important that this is easier to administer, thermostability, absolutely critical, and the single dose presentation really presents an opportunity to avoid missed opportunities to vaccinate. There will, we anticipate, be differences in countries' priorities based on their immunization program strengths and weaknesses. But this is really the beauty of this, this framework. Next slide, please. We also had the opportunity to validate the work that we've been doing with MMGH on vaccine use cases. You can see here that we had identified 6 vaccine use cases across the delivery setting and the context. And we evaluated these with the innovation framework. We were able to get good confirmation that delivery by healthcare workers in settings with limited or no healthcare services were an important use case for MR MAPS, which is what we anticipated. And that the delivery by healthcare workers presents an opportunity, but is really going to require a lot of work on the policy side, because at the present time, there is not a policy that will enable community healthcare workers to give vaccines. So that is something that's important for us now at this stage of development to begin to assess in terms of the broader potential use for this innovation if it becomes available. One of the use cases that has often been talked about for microarray patches is self-administration. But in the context of immunization program for infants and young children, which is what we were evaluating here, there was not a lot of confidence or comfort in discussing the self-administration context at this point in time. There would likely need to be more research and policy considerations in terms of thinking about self-administration. So getting greater clarity on these important use cases helps us with the demand assessment work that we have been doing, and we have also published a couple of papers with respect to that I won't go into today. Next slide, please. And finally, we did use these workshops as an opportunity to probe around whether countries would introduce MR MAPS and what would be some of the decision-making factors. Interestingly, Ethiopia told us that they would be really looking at a full switch from needle and syringe to microarray MAPS for routine immunization and campaign. So this again was a really unique learning for us, because we had made the assumption that this would really be about campaigns and it would be about house-to-house vaccination. But here we have a country that's saying that they would really be interested in using it for routine. And actually, if you look at the yellow little segment here, they were not interested in only using it for campaigns. And likewise for Indonesia, we learned that they would really be interested in partaking in a pilot, an implementation pilot, to look at the optimal implementation of this innovation. Next slide, please. So in terms of takeaways for MAPS, I think Phil said it at the very start, there's an urgent need for urgent public need for novel vaccine delivery innovations. Microarray patches offer a multitude of advantages and could be applied to multiple vaccines. So it really offers potential for true game-changing innovation. There are several vaccine maps in the clinic, but when we're talking about a vaccine map that is limited to LMIC use, it does present a challenging investment case, but we are doing some work on that now to really strengthen that investment case. Understanding the demand and market size is key for the investment case. And we have the tools and mechanisms that we're using to really understand at the country level, what are the opportunities for microarray patches with measles and rubella vaccine in particular. This could be applied to other vaccines and other innovations. And there's been a lot of interest from vaccine maps, so we're really helping to create a pull and demand for this innovation. Next slide, please. And really, just to say thank you so much to the many people who've been involved in these studies, particularly at WHO, the developers, our funders, the Bill and Melinda Gates Foundation, and the UK Foreign and Commonwealth and Development Office, and our partners at BIPS. Thank you very much. And I apologize for the tickle.
Thank you very much, Dr. Gersing, and I want to thank you for really kind of leading this effort on behalf of the WHO and bringing all these partners together and keeping this moving forward. It's a very exciting technology, and as you said, could really transform vaccine delivery. Our next speaker is Sir Adrian Hill, who's going to talk to us about also some very exciting work that's moving forward very quickly on the R21 Matrix M vaccine from the Jenner Institute. Sir Adrian, over to you.
Thank you very much. Could I have the next slide, please? Just a reminder that it's been pretty tough to make a malaria vaccine. This is the earliest report I know of, back in 1910, when we had two gentlemen in Algeria doing research on malaria vaccines. Next slide, please. So one of the reasons it's really tough is that you need exceptionally high titers of antibodies to protect against really any stage of the parasite's life cycle. And these are typically 10 to 100 times greater in terms of micrograms per mil than the antibody titers you need to protect against most other diseases. And inducing cellular immunity against the liver stage has proved no easier. Next slide, please. So the two leading malaria vaccine candidates, both of which have now had approvals, are shown on this slide. They are somewhat similar. They both target the circumsporozoite protein of the malaria parasite, which encoats the sporozoite, the form that the mosquito injects into the skin. Luckily, in the center of that protein, there's a run of repeats, 20 to 40, according to strain. And those are just 4 amino acids repeated many, many times. The earliest vaccine, going back to 1987, was RTSS, with repeats fused to the C terminus of the whole protein. And more importantly, fused to hepatitis B surface antigen, which forms a nanoparticle spontaneously. So you end up with a nanoparticle vaccine. Except that to make this nanoparticle, they needed a 4-fold excess of unfused hepatitis B surface antigen. So around about 90% of the vaccine is actually hepatitis B and 10% is malaria. In 2011, in our lab at the Jenner Institute, a graduate student, Kath Collins, succeeded in expressing in a different type of yeast, just the fusion protein. So if you, on the next slide, just depict how they appear, RTSS has little islands of malaria, as shown here on the surface of a Hep B particle, whereas the R21 vaccine has no exposed hepatitis B. You cannot stain it with a monoclonal antibody to hepatitis B surface antigen. Very briefly, the other difference between the two vaccines is the use of the powerful AS01 adjuvant from GSK, a proprietary adjuvant, with a TLR4 agonist as well as saponin, whereas we use a simpler matrix M adjuvant from the Novavax company that is simply the saponin without the TLR4 ligand. Next slide, please. Very briefly, we've done 4 important trials sequentially, starting with a challenge trial in the UK, where we showed that healthy volunteers immunized with 3 doses of this vaccine when challenged by mosquito bites in an established challenge model, showed sterile efficacy of about 65 to 82%, depending on exactly which immunization schedule you used. We progressed now in partnership with the Serum Institute of India, our commercial partner in this endeavor, to the coast of Kenya at Kilifi, where an age de-escalation study from adults to children to infants showed good safety and, interestingly, much higher immune responses in the Kenyan children than we had seen in UK adults. I'll briefly mention 2 trials that we've done for efficacy, VAC76, a phase 2b trial at Nanoro in Burkina Faso, and then a phase 3 trial, which the data from which has been submitted for licensure. Next slide, please. This is the phase 2 trial, which in the first year showed an overall efficacy of 76%, which was rather exciting. That was unprecedented in the field and indicated that you can reach the WHO goal of 75% efficacy over a year. If you give a booster dose at a year, a 4th dose if you like, in the 2nd year, the overall efficacy over 2 years was 78%. The booster did help, going from the 6-month efficacy here of 64 to 81%. But if you give no more boosters and follow for 3.5 years, you've still got overall efficacy of 66%. So we are recommending a 4-dose regime. Next slide, please. So we then moved on quickly to a phase 3 trial, which occurred largely during the pandemic in West Africa at multiple sites and also in lower transmission sites in East Africa. 4,800 children randomized 2-to-1 to the malaria vaccine or to a rabies control vaccine. And importantly here, we changed the age range, which was 5 to 17 months with the previous RTSS work, to 5 to 36 months to allow us to try and protect 1- and 2-year-olds as well. Double blind, obviously. And we divided the sites into those that were heavily seasonal, where the rains came for 3 or 4 months in the year, and most transmission came soon after that, and the more perennial year-round transmission of malaria, for example, in East Africa. Next slide, please. And in a nutshell, the primary endpoint was, well, 2 really co-primary endpoints at the seasonal sites. The overall efficacy over a year's follow-up, the first year was 75%, very tight confidence intervals here, as you can see. And at the standard sites, actually not significantly different, but a bit lower at 67%, and not very different at the different sites. Next slide, please. That was the per-protocol analysis in the overall population of children. Interestingly, if you study just the 5 to 17-month-olds, as shown here on the top line, the efficacy does look higher than in the older children, not massively by 9%, but you're up to 80% overall, and that's 79% at seasonal sites and 82% at standard sites. And that's probably important because this is the age group we're actually most likely to vaccinate in the coming months and years. And interestingly, I won't mention immunology much in this talk, the antibody titers are higher in the younger children than the older ones. Next slide, please. There was a lot of discussion after our phase 2 trial as to whether the higher efficacy might be related to the seasonal setting. The answer is no. The efficacy is not any different between the seasonal and standard sites over the first year. We now have data out to 18 months, and that seems to be holding up well. Next slide, please. And importantly on safety, so we have data on now over 3,000 children given 3 doses and now 4 doses of this vaccine. There's no adverse event of concern, certainly no serious adverse events that are overrepresented in the vaccinees compared to the controls. You've got 2-to-1 randomization here, except possibly febrile convulsions. The numbers are small, but we had 5 in the malaria vaccine group and just 1 in the control group. You would expect 2-to-1, and that was a side effect seen with RTSS, which also has a saponin adjuvant component. Next slide, please. But these are all self-limited. Are we doing better than RTSS? Well, in several ways, we believe so. Just on efficacy, this is in the 5 to 17-month-old groups. in the phase 3 of RTSS, 55% as reported several years ago, and we seem to be significantly higher. Next slide, please. Immunologically, we think we have a basis for that. We're seeing very good, very high antibody titers with our nanoparticle in adjuvant. These drop more slowly than has been reported for, say, RTSS. The trough after a year here is has not dropped as much as with RTSS. We can boost very well. And even out here at the second and third year after vaccination, we've maintained good antibody titers, which correlate well with vaccine efficacy, as we've published. Next slide, please. Briefly on deployability, you were hearing about issues like this a moment ago. There are many doses of R21 available now. We have worked on a single vial presentation. Other vaccines use vials that have to be mixed at the bedside or in the village, if you like. And the single vial presentation looks fine, similar to the mixed formulation. We have 24 months at 2 to 8 degrees, and indeed 40 degrees for 2 weeks, which surprised me. That is very good indeed for a field distribution. Serum Institute of India, of course, are the largest vaccine supplier to Africa. They'll be able to make up to 200 million doses from next year, but quite a lot of doses this year as well. And multi-dose vials are coming very soon. Next slide, please. So just before acknowledgements, I will mention that in the last couple of weeks, 2 West African countries, Ghana and then Nigeria, have approved this vaccine for use in 5 to 36-month-olds, and we anticipate there will be others very soon. And we're looking forward, hopefully, to pre-qualification and that policy recommendation by WHO fairly soon too. Thanks to my colleagues at the Jenner Institute leading various aspects of this phase 3 trial. Of course, to the Serum Institute of India, Umesh, and many colleagues there. And on the last slide, most important of all, the PIs who did most of the work at these trial sites in West and East Africa. Thanks for your attention.
So Adrian, thank you very much. This is such an exciting development. We've waited so long for a more highly efficacious malaria vaccine, and it's so exciting to see this really move forward and to see the results of your phase 3 trials. So thank you.
Thank you.
Our third speaker is my colleague, Professor Ruth Karen from the Johns Hopkins Bloomberg School of Public Health. And Dr. Karen's going to speak to us about new developments in vaccines for respiratory syncytial virus. So Ruth, over to you.
Thank you very much, Bill. And it's really a pleasure to be with all of you. 2023 is very much has already shown to be and is going to promise to be very much the year of RSV in terms certainly of licensure and registration of new products and hopefully deployment of the same. So for those of you who don't know a lot about RSV, it is the leading global cause of pneumonia in young children. Estimates, the most recent estimates based on meta-analyses are 33 million cases of lower respiratory tract illness every year, 3.5 million hospitalizations, over 100,000 deaths. And very importantly, although RSV infects all children in every country in which it's ever been studied, the vast majority of RSV deaths occur in low and middle income countries. As you'll hear, RSV is also an important cause of lower respiratory tract illness in older individuals, we know in high income settings. I've given you here the estimates for hospitalizations and deaths in adults living in the United States. We don't have data that's nearly as robust in low and middle income countries. As you can see, the estimates of hospitalization and deaths are probably vast underestimates given the data that we know from the United States alone. Next slide, please. So just a few words about the overall strategies for preventing RSV, lower respiratory tract illness. For the very youngest babies who are most likely to experience hospitalization or death, the strategy has really been around passive immunization to protect these infants with antibody in one of two ways. Either to immunize their mothers when they're pregnant and allow for transfer of antibody from mother to infant, or to immunize infants themselves with a monoclonal antibody that's been modified in the FC portion to allow a longer half-life. So these are so-called vaccine-like monoclonal antibodies. A single dose will last for about 6 months. The strategies, then we think, so that's one population. A second population are older infants and toddlers. For those children who may not experience as frequent hospitalization or death, but are really still at increased risk of serious disease. And there's a larger population of them than there are of infants 0 to 6 months of age. Live attenuated vaccines are in development, as well as mRNA vaccines. And then finally, RSV F subunit and vectored vaccines are being developed for older adults. I will just, in the interest of time, I have not gone into the structural biology of the RSV fusion protein, but I just wanted to say that a lot of the innovations that you'll hear about in the next few slides are really the consequence of the landmark work that Barney Graham and Jason McClellan did to understand the structure of the RSV fusion glycoprotein, which is the major protein that gives a protective antibody response, and understanding that in the pre-fusion conformation, this protein is highly immunogenic and gives you good neutralizing antibody, but in the post-fusion conformation, which is what you get unless you take pains to stabilize the fusion protein, it's significantly less immunogenic and you get less neutralizing antibody. So next slide, please. I just wanted to point you to a public service that PATH provides. This is a chart that's updated about every 6 months or so by PATH that lists all of the RSV vaccines and monoclonal antibodies in development. The URL is at the bottom of the slide. And what I'm going to spend most of my time talking about today are the two products that are outlined in red, the Pfizer RSV-F maternal vaccine and the nirsevimab, which is a long-acting monoclonal developed by AstraZeneca and marketed by Sanofi. Shown in yellow are other products that I will touch on, 2 products developed for immunization of the elderly, and then also an RSV monoclonal by Merck that is in late-stage phase 3 development. Next slide, please. So I'm going to spend, this is my only slide about RSV vaccines for older adults, and this is really in the interest of time. And I will say that it doesn't really in any way do justice to all of the work that was done to develop these vaccines and to test them in adults. As you can see, these are extraordinarily large efficacy trials. But there are 2 products that are really have been put forward for licensure or registration. One from GSK with a study that enrolled about 25,000 participants. The efficacy against any RSV acute respiratory illness was over 70%, and then against lower tract illness was almost 83%, and huge efficacy against severe illness, as you can see. The Pfizer product was evaluated in 36,000 participants, and you can see everybody has slightly different criteria for their case definition, but they're not identical, but they're certainly similar. What they showed is that the efficacy against lower tract illness was about 67%, and then with 2 symptoms and with 3 symptoms was about 86%. One of the differences I should just point out between the GSK and Pfizer trials was Pfizer had a predominance of, if I can call them this, the young elderly, meaning individuals between 60 and 69 years of age. About 2/3 of their participants were in that age range, which may be why they weren't able to show even greater efficacy against the more severe disease. These products are moving forward, have had favorable reviews by the US FDA Advisory Committee, and they're awaiting final determination from the FDA. And certainly these are in review by other regulatory authorities. Next, please. So I'm next going to talk about the maternal RSV pre-fusion protein vaccine. And I will say this is a product, this is made by Pfizer. This product is identical to the product that they evaluated in older populations. This product was studied in 18 countries with about 7,300 participants. What you can see is, although there is geographic distribution of these countries, there are relatively few enrollees from the global south, and in particular on the African continent. South Africa and the Gambia were the only two countries that enrolled children, enrolled pregnant women. And women were vaccinated when they were 24 to 36 weeks gestation. Next slide, please. So these are the results of this study, and I think we can probably click again because it'll outline the vaccine efficacy. So there are two endpoints looked at. One is medically attended lower respiratory tract illness caused by RSV. And what you can see is that the efficacy here is between 50 and 60%, and importantly, is sustained out to 6 months, 180 days after birth. One thing I should just mention, which is it is what it is. I don't think it's particularly consequential, is for that 90-day endpoint. The lower bound for success was preset at 20. So technically, the lower bound was not met for that first endpoint at 90 days after birth, probably just owing to a numbers issue. Certainly it was meant for every other time point. And if you look at severe medically attended lower respiratory tract illness, you see increased efficacy ranging from about 70 to about 80%. I did just want to make one comment about the conduct of these trials and also the nirsevimab trials that you'll hear about in a moment. I think that the sponsors, the investigators, and the participants were absolutely heroic in this. Because not only were these trials done during COVID, which presents its own logistic barriers, but RSV seasons were completely disrupted and altered by the COVID pandemic, such that it was really very difficult, particularly in these maternal immunization trials, to think about the timing of vaccination, relative to the predicted RSV season and relative to the actual RSV season. So to have been able to successfully conduct these trials is really a major accomplishment. Next slide, please. So maternal RSV pre-F has been submitted to the US FDA. The advisory committee review is on May 18th, and I will tell everybody that these are live streamed and open to the public. So those of you who are interested should dial in. Submission to other regulatory authorities is in progress. In the US, the challenge is, and I think this may apply to other high-income countries, we don't do that well with maternal immunization, especially compared to childhood immunization. Our rates for Tdap and influenza are on the order of 55 to 60%. The second issue that has arisen in terms of global implementation is around data that are available from low and middle income countries. And it's not really the issue of whether we think these products would work in those countries. We think that in terms of mechanism of action and amount of antibody generated, that it is likely that they would work. But the question is really, what is the full benefit of this vaccine? And have we done enough? You know, when countries are faced with difficult decisions about a multiplicity of vaccines that may be available for implementation, what information is needed for them to make decisions? So next slide, please. And there is some work. There is a group that the WHO is convening that I've had the fortune to be on, that really is about helping to design studies to assess the full impact of preventing severe RSV disease in infants and children. There are potential short and long-term impacts on the well-being of the individual child, the family, and the health system. Certainly impacts on all-cause pneumonia, on all-cause infant hospitalization. And this is a particular issue in LMICs, and that's because in countries, and we've heard wonderful presentations on this recently about the experience in Bangladesh. What happens in many countries where there are bed shortages is it's first come, first serve. So in an RSV season, when many babies are being admitted for RSV and for oxygen administration, it may be that other seriously ill infants and young children are being turned away from hospital because of bed shortages. So it's not just the effects on RSV hospitalization and mortality, it's really on all-cause infant hospitalization and mortality. Then effects on antibiotic use in infancy, and something that may not be able, certainly won't be able to be immediately ascertained, but long-term lung health is also a consideration. There are multidisciplinary and innovative work streams that are going to be needed to assess, quantify, and communicate these impacts. Next, please. So a couple of words about vaccine-like long-acting monoclonal antibodies. There are 2 products in late-stage development, and one has been approved in a few countries, nirsevimab, which is what I will really spend our time on, and clesrovimab, which has been developed by Merck, is also a very promising monoclonal antibody with a phase 3 trial in progress. Next, please. So these are the data for nirsevimab. This was actually published in the New England Journal in the same week that as a letter. This was a follow-up to their initial study at the same time that the data that I just spoke to you about from Pfizer were published. This is efficacy against medically attended lower respiratory tract illness, RSV lower respiratory tract illness with hospitalization and very severe disease. And you can see high efficacy against all of these endpoints. Unlike the Pfizer trial, the nirsevimab trial was actually able to show efficacy against all-cause lower respiratory tract illness with and without hospitalization, around 38%. Next, please. Yes. And this is, I'm sorry, could you go back, please? Thank you. The number needed to treat to prevent one all-cause medically attended lower respiratory tract hospitalization was 53. And there were 57 days of hospitalization averted for every 1,000 infants treated. Next, please. So this product, nirsevimab, has been approved by the European Commission in October, was approved by UK Medicines and Healthcare Products Regulatory Agency, the MHRA, in November, and was approved by Health Canada just last week. It's been submitted to the US FDA with review expected in 2023, and it has been submitted to other regulatory authorities. The use of this product is actually anticipated toward the end of this year. Next slide, please. So we're starting to think about and talk about the beginning of the end for RSV disease in children. There have been incredible advancements in the past decade. As you've heard, I hope, and agree, there are two really excellent products that have been developed for passive protection of the young infant, with a third likely in the next few years. Affordability, access, and acceptance are going to be key determinants of success, as will determining and communicating the full value of RSV immunization. 95% of RSV deaths occur in lower middle income countries. I started my talk by saying that. We need to do all that we can to ensure global use. I will say that the RSV pre-F maternal vaccine, there are plans for global deployment. Among other things, the Gates Foundation has provided funding for Pfizer to develop multi-dose vials for use specifically in LMICs. We have not heard anything to date from Sanofi about plans for global distribution of nirsevimab. Progress is accelerating. I didn't have time to talk about it today, but things are really moving forward in terms of development of infant and toddler vaccines, which I think will be needed to more completely address the burden of RSV disease in children. These products that I've just told you about, I think will do a huge amount if they are deployed to take care of children, the very youngest infants, 0 to 6 months of age. But we certainly need products for older children. Next, please. I just wanted to end, and my acknowledgements, unfortunately, are not on this slide, but particularly I just wanted to acknowledge Tanya Villafana from AstraZeneca and Iona Munjal from Pfizer for sharing their slides with me. Thank you all.
Great, thank you, Professor Karen. And it's again exciting to see here a number of different products for a number of different target audiences or target populations. I just want to remind all of our viewers that there's a Q&A chat that you can put your questions into, and we'll see how much time we have left at the end and take some of your questions. But please use the Q&A chat for questions. Our next speaker is again another colleague of mine, Dr. Kausar Talat, here at the Johns Hopkins Bloomberg School of Public Health and the Center for Immunization Research. And Dr. Talat's going to talk to us about innovations in Shigella vaccines. Kausar.
Thank you very much, Dr. Moss. Next slide, please. So I'm going to start with a little bit of background on Shigella. Shigella is the second leading cause of diarrheal deaths worldwide, and it is the most common bacterial cause of moderate severe diarrhea in children under 5. And in the second year of life, so from 12 months to 24 months, it is the most common cause of diarrhea in children all over the world. It's estimated that nearly 200 million cases occur worldwide, with over 200,000 deaths, about half of them occurring in children. But in addition to causing diarrhea illnesses and deaths, Shigella also contributes to the loss of growth faltering, linear growth faltering in children. It contributes to malnutrition and stunting, which puts children at increased risk for additional infections and potentially greater morbidity and mortality in the future. Because of this, the WHO has prioritized Shigella for vaccine development, and it's a very exciting time to be in Shigella vaccines. So there's been a lot of progress. In addition to the infections in low and middle income countries in children, Shigella also causes diarrhea in travelers because it requires a very small dose of bacteria in order to cause illness. Enough bacteria that can fly on a fly, for example, can transfer Shigella and make people sick. And we've also seen in high income countries outbreaks in daycares and in populations of men who have sex with men. Next slide, please. Shigella is an invasive gram-negative bacteria, and the invasion depends on virulence plasmids that are transmitted along with the bacteria, and the bacteria enter and destroy the cells of the intestines, leading to the illness as well as to bloody diarrhea or dysentery. There are 4 species that are divided into over 50 serotypes based on the O antigen, which is the surface portion of the lipopolysaccharide that is on the surface of the bacteria. The species are listed below: Shigella dysenterii, flexneri, boydii, and sonii. And it's really flexneri and sonii that cause the majority of disease worldwide. Dysenterii causes large epidemic outbreaks of Shigella. Dysentery, however, we haven't seen that as much in recent years. And unfortunately for vaccine development, immunity to Shigella is felt to be serotype specific. So if you've been infected with one serotype, you're protected against and future infections with that serotype, but not against other serotypes. Next slide, please. We all know that that Shigella causes diarrhea, but it also causes dysentery, which is bloody diarrhea. In addition, people who have Shigella generally feel quite ill. There's a lot of fever and abdominal pain, as well as other associated symptoms. These generally last about a week or less, and the infection is generally self-limited. However, there are significant sequelae from having shigellosis. Diarrheal infections lead to stunting and malnutrition in children, and you have both physical stunting as well as cognitive deficiencies. And children who've had repeated diarrheal infections early in life have been shown to enter school late, leading to overall decreased time in school and decreased future earning potential, further perpetuating the cycle of poverty that occurs in many of the places where this infection is endemic. Next slide, please. In addition to causing infections, Shigella is a rapidly emerging antimicrobial resistance threat. Like other gram-negatives, there are antimicrobial resistance genes that are transferred on plasmids between the bacteria. And there has been increasing cases of extensively drug-resistant shigellosis that have been reported from all over the world, including low and middle income countries as well as high income countries. In the United States, we've seen an almost exponential increase in extensively drug-resistant shigellosis. This led the CDC to issue a health alert, a health advisory alert in February of this past year. And this is one of the other reasons why this has been a target for vaccine development. Next slide, please. However, there have been significant barriers to vaccine development that have stymied Shigella vaccines over the last 50 or so years. The main one is the diversity of Shigella serotypes. As I mentioned, there's over 50. However, many of these serotypes are uncommon, and epidemiological studies done in the GEMS, MALI-D, and NALVIDA cohorts throughout over the last 15 or so years have shown that Shigella flexneri 2A, 3A, 6, and sonii cover 75% of global strains. And so if these are included in a vaccine as a quadrivalent vaccine, then you'll cover the majority of Shigella strains. We know that oral vaccines have less efficacy in low and middle income countries. This has been shown with polio as well as with the rotavirus vaccine. It's likely to do with environmental enteropathy and other co-infections. And so a lot of the leading Shigella candidates are going to be given through injection rather than vaccine candidates, rather than orally. We have not yet been able to identify a correlate of protection for Shigella, despite a tremendous amount of work in this area. And it is likely that the O antigen is an important target, but also the surface proteins that are shown in the figure on the right, which are important for the invasion type 3 secretion system that gives Shigella its virulence, are also important antigens for protection, and some vaccines will include these antigens in order to increase the protection provided by the vaccine. Because this is a disease that is primarily affecting people in low and middle income countries, any vaccine developed against Shigella needs to be affordable, and there's very little commercial incentive for vaccine manufacturers to focus on Shigella because the return on investment is not as high as with other vaccine targets. And there's ways to overcome this, and we'll talk about that in the coming slides. There have been many Shigella vaccine candidates that have been tested over the years that are either too reactogenic, and so development has been stopped, or they have not been effective. And what we found with some vaccine candidates is there's significant differences in immunogenicity and the efficacy of the vaccine when tested in high income versus target low income settings, or in adults versus infants. And the previous generation Shigella vaccine worked really well in trials in adults and older children, and not so well in target populations. Next slide. So the WHO has released some target product profiles for Shigella vaccines. Dr. Gersing was instrumental in the development of these profiles, and the target populations are infants and young children in low and middle income countries. Travelers are also another target, and we'll talk about that as our military personnel. In addition, it might be appropriate to use a Shigella vaccine in children who live in crowded communities in middle or high income countries where Shigella is endemic, or there have been outbreaks, immunocompromised children who are at risk for more severe disease, men who have sex with men where there have been seen outbreaks and where the extensively drug-resistant Shigella is circulating, people who are living with HIV, and older children and adults in LMICs may be vaccinated on the private market, which might help support the financial aspects of the development of the Shigella vaccine. Next slide, please. Dual market for Shigella vaccines can help with the commercial appeal of this. So the children in low and middle income countries, especially young children 6 to 12 months of age, are the WHO target group. For the development of these vaccines for this population, there will be required a phase 3 field efficacy trial in this population for licensure, and because of the failure of previous vaccines in this population. And so there are ongoing age de-escalation trials in this population as we speak to try to look at the safety, immunogenicity, and hopefully efficacy of leading vaccine candidates. And also, again, to improve the commercial appeal and also the acceptability of these vaccines, it might be good to have a combination vaccine targeting other diarrheal pathogens or even other pathogens to decrease the number of different products that are given in this highly immunized age group. The other target population are travelers who are mainly from high-income countries. They can pay more for vaccines, and they can offset the price of the vaccine to low and middle income countries. And for this population, we're actively looking at using controlled human infection models or challenge studies to support licensure in this population. And also for this population, combination vaccines may be better. Next slide, please. The first Shigella vaccines were tested in the early part of the 20th century, but we still don't have an effective vaccine. There have been an explosion recently of Shigella vaccine development over the last 20 or so years after the initial proof of concept in the 1990s, where Dr. Robbins and Dr. Schneerson had a conjugate vaccine that was, again, very efficacious in adults and older children, but not so efficacious in younger children. Next slide, please. And this is now the Shigella vaccine landscape that is currently in development. There is one vaccine that is a conjugate vaccine that is phase 3 trials in China. There are 3 vaccines that are all in phase 2 and late phase 2 trials as quadrivalent vaccines. 2 of them are conjugate vaccines, which we'll talk about on the next slide, and one is a membrane blood vaccine called Gemma from GSK. The 2 conjugates are from the Pasteur Institute and LimaTech. There are live attenuated vaccines in development that will be administered orally. And there's also inactivated toxin vaccines that are also currently in HD escalate or going to be in HD escalating trials shortly. Next slide, please. The conjugate vaccine technology seemed to be the leading technology, so I wanted to explain a little bit of what this is. This is isolating the LPS or lipopolysaccharide of the bacteria, purifying the O antigen, and then conjugating it to a protein. That protein can be tetanus toxoid, it could be Pseudomonas exoprotein A, but anything that will allow that polysaccharide to be more immunogenic and to induce a T cell dependent immune response, which is a longer and more durable immune response than giving a polysaccharide vaccine by itself. The 2 candidates, the leading candidates that are using this technology are the LimaTech Shigella vaccine, which is a 4-valent vaccine that uses an E. coli expression system to do the conjugation through bioconjugation, as well as the Pasteur Institute, which uses a chemical conjugation pathway to do this conjugation. Next slide, please. I'm going to end here and say that Shigella is a major cause of morbidity and mortality from diarrhea and a threat to people in low and middle income countries, as well as travelers, and as well as populations within high income countries. And so a vaccine for Shigella is a priority to prevent morbidity and mortality and to decrease antimicrobial resistance. It's been almost 100 years since the first vaccines have been tried, and there's several promising candidates that are in clinical trials. They're not as far as the vaccines that we've heard about this morning, but I think in the next 5 to 10 years, we're going to see very promising news on the Shigella vaccine front. Dual markets possible to help with the feasibility of this vaccine. Next slide. Thank you.
Great. Thank you very much, Dr. Talat. And you really highlighted some of the challenges in developing Shigella vaccines, but also some of the challenges with the investment case and the target populations and the diverse target populations. All right, we're going to move on to our final speaker, Dr. Rupali Lemay, who is the Deputy Director here at the International Vaccine Access Center and Associate Professor at the Johns Hopkins Bloomberg School of Public Health. And she's going to talk to us about progress with vaccines for tuberculosis.
Thank you so much, Bill, and thanks for letting me participate in this really exciting webinar. I know we've had a lot of really great speakers, and I know there's a lot of questions, so I'll keep my remarks relatively short here and talk about our TB vaccine progress and where we are. So if we can move to the next slide, what I'm going to quickly talk about is the case for a new TB vaccine. I'll spend just a little bit of time and talk about current vaccine that we currently use for tuberculosis, which is the BCG vaccine. A little bit with regards to what are some challenges when we think about TB vaccine development and some key vaccine candidates, because there's quite a few that are in the pipeline. Finally, I'm going to end with really talking about what are some implementation considerations, because once we actually do have a viable product that's pre-qualified, how do we actually get it rolled out and into arms so that individuals are accepting the vaccine? So if we move on to the next slide. Yes, we can go on to the next one. So as I mentioned, the current vaccine that we use for tuberculosis is called BCG. And what do we know about it, right? So this is actually a vaccine that actually is quite, has been around for quite a bit. So it was first given for protection almost 100 years ago, actually a little over 100 years ago. And it's still the most widely used vaccine and oldest in use. You can see some of the numbers here. More than 4 billion children have been vaccinated using this vaccine, and more than 150 countries still recommend this BCG vaccine for infants. However, I would argue that there are some limitations when we think about the BCG vaccine. A number of studies have come out that have essentially shown that when we look at effectiveness of the vaccine, we can see actually, when we think about household contacts, that there's only about an 18% effectiveness against TB. And again, you can see it's slightly higher in those that are younger ages, i.e. less than 5 years of age. This vaccine does confer and does lead to a 75% reduction in death, but that's only through age 15. So as individuals are older and are exposed to tuberculosis, this vaccine is not as effective with regards to preventing death. The other key issue, as many of you know, a key comorbidity related to TB is if an individual is infected with HIV. And one of the key things that has recently been discovered is that there is reduced safety in HIV positive persons, and as a result, it's not recommended. The last piece that I would recommend is that although we're talking about effectiveness, one thing to think about is that it has very limited impact when we talk about transmission itself. So these are just some of the key factors and limitations related to the BCG product. If we move, thank you. So why do we need a vaccine now, right? So what is really the reason why we should think about investment? We should encourage investment, and we should also think about how countries can prepare for new TB vaccines. So WHO has put out the End TB strategy, and by 2030, there are key milestones that need to be hit. So the argument here is that by 2030, new TB cases should be reduced by 80%, that deaths should be reduced by 90%. Now, without immediate action, if we don't have enough R&D investment into vaccines or other therapies, by 2030, what we do know is that 43 million will develop tuberculosis. More than 6 million will actually die. And in terms of economic costs, it's quite hefty. It will cost approximately about $1 trillion. And so as a result, when you look at the figure on the right, when you look at the current global trend of what's happening, it's really important for us to be able to introduce new tools. This includes vaccines, of course, but it also includes new drugs and treatment regimens so that we can think about active as well as latent TB, as well as a point of care test. So that's why it's so important and essential for reaching these NTB 2030 targets. Next slide. Great. So what are some of the strategies when we think about tuberculosis, right, in terms of a vaccine? So there's really 3 ways and 3 endpoints that you can think about any sort of TB vaccine candidate. So there can be prevention of infection, which is POI. There's prevention of disease, which is POD. And then there's therapeutic and prevention of recurrence, right? So there's a number of different ways that we can think about where perhaps treatment as well as vaccines could actually intercept and really think about interrupting one of these pathways, if you will. So if we go to the next slide, there are some key challenges, and I'm going to quickly touch upon with regards to TB vaccine development. So along the bottom, you can see essentially the phases through clinical discovery. So from discovery to preclinical through the phases. So phase 1, 2A, 2B to 3, and then post-licensure. There's been a real challenge to better understand what are the human protective immune responses to TB. And that really slowed down the discovery phase quite a bit. The second key challenge is that there really has been a lack of evidence related to animal models that predict protection in humans. In addition, there has been a lack of clear, I would say, correlates related to protection. And then more importantly, there is a need to really enroll a large cohort of individuals in any of these trials, i.e. more than 10,000 participants. It also includes a pretty long follow-up time, at least 3 years. So this has also slowed down vaccine development. Finally, one key thing, if you think about the global health priorities and how funding is allocated by different donor agencies, there really is a lack of international agency in terms of funding priority when you really think about the morbidity, mortality related to tuberculosis, as well as burden of disease. It really is disproportionate. So we can move to the next slide, please. So I didn't want to go through each of these because, as you can see, there's a huge number of vaccine candidates that are currently in this pipeline. So you can see under phase 1, there's several under phase 2. The ones that I think we're most interested in right now are, of course, the phase 2B and the 3. And the way that this was a slide that was actually developed by the Stop TB Partnership, where you can actually see the different populations that are targeted. A key thing to remember here that while really in terms of a vaccine strategy, that a lot of the strategy the priority focus has been thinking about adults as well as adolescents, we know that there are groups of individuals that have comorbidities that we really need to be thinking about. For example, those, again, that are HIV infected. One omission that I think continues to arise in these discussions when we're talking about TB vaccine candidates is that currently there are no candidates under development looking at pregnant and lactating persons, for example. So this is a key omission as well, and there's other target groups. You can also see essentially where the trials are and what the primary candidate indication is, meaning on that slide that I just showed you a couple of slides ago, whether or not the vaccine would prevent infection, whether it would prevent disease, or whether it would really prevent recurrence. So these are important things. So you can see that there's quite a bit in the pipeline coming up. Next slide. So in terms of implementation, as I mentioned, there's a number of candidates that are particularly in phase 2 and 3 that we're excited to see results from. However, one thing that I would argue that's really important at this point in time is that now is an opportunity to really build demand for TB vaccines. There's not a TB vaccine candidate on the market. The slide that I just showed you through the Stop TB partnership, our understanding is at most we might see some trial results in 2023 on a phase 3 candidate. However, that means that we have a little bit of time before a TB vaccine candidate will be pre-qualified and actually on the market. So as a result, this is actually the opportune time to think about this idea that we know that vaccines don't save lives, but vaccination does. So how do we actually restore vaccine trust? Because we have seen a decrease in trust towards science, as well as a decrease in trust in vaccines, given COVID and all of the other misinformation and science denialism that is happening. But I think most importantly in this space is that to prepare for TB vaccines, behavioral and social data collection is really critical, and thinking about how do we put the community at the center of these types of efforts will really help prepare countries once a TB vaccine is available so that individuals are interested in the vaccine and that they're able to access the vaccine. So if we move to the next slide. One thing that I will argue, though, is that building demand and confidence in new vaccines is very complex, just like in any other vaccine. So it's important when we're thinking about implementation considerations is to not only think about those that are affected by tuberculosis, but to think about their caregivers and their communities that surround them. This includes, by the way, healthcare workers who might have hesitation towards specific vaccines, so that when a vaccine is available, they'll recommend it and they're able to do this with confidence. In addition to that, it's important to think about and engage with vaccine manufacturers now. Of course, the key vaccine alliance partners, different ministries of health at the country level, but as well as other immunization advisory boards, so that when they're thinking about the evidence and looking at the evidence, they will make a recommendation that is supportive of a new TB vaccine to be included into an EPI portfolio. I think if we move on to the next slide. Yes, so just a few conclusions that I want to make here. As I mentioned, if we do nothing, we will see more than 6.6 million people will essentially die from tuberculosis by 2030. Vaccines are a critical piece of this pie. So having a safe and effective and affordable vaccine, by the way, is crucial if we really want to reduce morbidity as well as mortality related to TB. As I showed you, there's several challenges that have really hindered our vaccine development process. Again, we're very hopeful that some of these trials that will start showing results, hopefully later this year, that we actually see a strong candidate that has strong vaccine efficacy as well as safety, because that's going to be critical. However, as I mentioned, given the current status, an introduction of a new TB vaccine is likely a few years away, likely 2025 at the very earliest. And so as a result, it's really important to start to get countries ready now so that when a vaccine is available, not only are health systems ready, but individuals are ready to accept it. And I think most importantly, really beginning to think about how we can continue to build that trust between community engagement and stakeholder engagement will be very critical, because again, vaccines don't save lives, but vaccination does. And I think those are the end of my slides. So I'll turn it back over to Bill. Thank you.
Yes, thank you, Rupali. And those lessons around country readiness and demand generation are really important for all the products that we've talked about today. So we do have some questions that have come in through the chat. And we have a few minutes left, about 10 minutes left. So what I think I'll do is try to group them, but and also encourage our listeners that if there are additional questions you want to add to the Q&A chat, please do now. So, Dr. Gersing, maybe we'll start with you on the microarray patches. And there were a couple of questions that came in, and I'll just kind of group them for you. One is just a little more information on the logistics of how these are actually administered. How long do they stay on? Are they solid needles or self-dissolving needles, particularly the products that are further in development? There was a second question about, I'll kind of reinterpret it, about the potential role for MAPS in the global polio eradication initiative and a role there. And then there were two questions really around, specific country use, one in possibility for a vaccine trial in Maharashtra, India, and any commitment from the government of Indonesia on rolling these out, because that's where the capacity workshop was held. Those may be more difficult ones to address, but maybe we could start with just some of the logistics of administering the MAPS and potential use in the global polio eradication initiative.
Sure, thank you very much. So the MAPS themselves are, as I mentioned, they're pretty small. They're about the size of a coin, with a covering that you peel off and you press onto the skin. There are two types of MAP that are in development for MR that I'll focus on. One has coated needles, so the solid coated needles with an applicator. So you put it on the skin and you press, and the vaccine dissolves off the coated needles and into the skin, and the wear time for those is a few seconds. The alternative is a dissolvable type of patch, and that's without an applicator, and it looks like a plaster actually, but it's a similar kind of mechanism where you press it on the skin, and those are up to 2 to 5 minutes to allow the vaccine to dissolve into the skin. Then you peel it off and you dispose of it. So it really is a very sort of minimally invasive procedure. And of course, one of the things that we're looking at in the studies that we do is what is the level of comfort with little kids when they sit with this thing sort of on their arm, and how do we keep them entertained? So that's really on more of the logistics pieces, and also so the two types for measles and rubella are coated and dissolvable. With respect to country interest in vaccines on MAPS, this is really the whole purpose of the capacity workshops. And we're really hoping to create demand for these innovations through doing workshops in different countries and articulating how these can be used and the barriers that they would overcome in the measles and rubella immunization programs. So I think the jury is still out, but certainly I've put the details of the manufacturers into the chat for anybody to reach out to engage with them in terms of their clinical development plans.
Great, thank you, Dr. Gersing. And one question just came in on the potential for combination vaccines using MAPS, say the measles, rubella. Yep.
Yeah, I think that's something we are all very interested in. Measles, rubella is already a combination. We could be limited though by the number of needles on the patch and keeping the size small and how many vaccines we can actually load, but certainly we're looking into that. I didn't answer the question about polio. That would also need to be a combination. We have, not we personally, but there has been work done on polio. The challenge has been actually stabilizing the vaccine antigen on the MAPS, but agree it's an incredibly important target.
Mm-hmm. Great, thank you. Sir Adrian, there was one question on the malaria vaccines, and you addressed this somewhat in your talk, but I think many of us are aware of the limitations in the supply of the RTSS vaccine, and I think the potential for a much greater supply of your R21 vaccine. And maybe you could just talk a little bit about it. And I know you mentioned it, but your vision for kind of the rollout, say, of the R21 vaccine. And you mentioned the two countries, the Gambia and Nigeria, that have already approved the vaccine. So it's moving quite quickly. We'll just need to unmute.
Apologies, I was muted. Yeah. So, our vision is that anybody who wants this vaccine can have it. To facilitate that, we've done a couple of things, partnered with the world's largest vaccine manufacturer. They had already worked with Novavax on a COVID vaccine, and very large numbers of doses of the adjuvant from Novavax have been used worldwide, providing very useful safety data. Very importantly, we wanted the price of this vaccine to be affordable. That's not defined yet, but we're thinking $3 or $4 a dose, maybe. That's for the company to decide, but it won't be $10 or more as RTSS is this year. I understand there'll be 4 million doses of RTSS available in 2023. This year, we may only have 50 million or thereabouts, but next year, hopefully it'll be, well, whatever is needed, 150 million, 200 million. But importantly, we envisage other applications for this vaccine than the 5 to 36-month-old age group. Clearly, there's a need for vaccine in adults, particularly women planning to become pregnant. We're already working with partners who are undertaking or about to undertake trials of elimination in low transmission areas with the vaccine. And importantly, we have a development pipeline at the University in Oxford. My colleague Simon Draper has a blood stage vaccine. We're combining that with R21, hopefully to get even higher and more durable efficacy. And of course, with transmission blocking components that you would include in a transmission blocking vaccine. So we don't see this as the final solution. We see this as an important step forward, decreasing the cost and increasing very substantially the availability of doses.
Great, thank you very much. And Professor Draper was just here participating in our World Malaria Day event here at the Johns Hopkins Bloomberg School of Public Health, so talking about the vaccine. Wonderful.
Perfect.
Maybe just in the last few minutes, we won't get to all the questions that are now coming in, but maybe for Dr. Karen on the RSV vaccines, there was a question about Guillain-Barré syndrome as a adverse event, potential adverse event. If you could comment on that. And then maybe an interesting one about the approach in low and middle income countries, kind of the trade-offs between maternal immunization and long-lasting antibodies for newborns.
Okay, yeah, and I saw one other one, which if I talk fast, I might be able to address all three. So first, around the neurologic issues, it is true that in the Pfizer trial that had about 17,000 vaccinees, there were 2 neurological adverse events that occurred. One was Guillain-Barré syndrome. One was a related syndrome called Miller-Fisher syndrome that occurred in an individual with a lot of other complex illnesses. In the GSK trial, there was one instance of Guillain-Barré syndrome in about 12,500 vaccinees. These are, these are events to pay attention to, but I don't think you can say anything about the incidence that one might or might not expect based on these isolated events. Certainly, these are issues that the companies will address in post-licensure, post-registration studies with larger populations to look at this. I will just say that in the maternal vaccine trial for Pfizer, there were no such neurologic events reported, which I think is a bit reassuring, but certainly to be followed in phase 4 trials. Long-acting monoclonals versus maternal immunization and LMICs. So it's my position that just as I think that there are countries, I don't think one size fits all. I think there are countries with strong antenatal care systems where maternal immunization might be used or might be used by countries to strengthen their antenatal care system. I think there are alternatively low and middle income countries that have strong EPI programs that would prefer to give this through EPI. My own personal view is that just as high income countries will have a choice that they can make, LMIC should also, and I can just say that there's a lot of work going on around making affordable monoclonal antibodies. I've been at meetings about this, and RSV is high on the list because we know exactly the antigens that are relevant and necessary. And just to answer the person quickly who asked about why don't we use subunits in older children, I didn't have a chance to talk about enhanced RSV disease and concerns around that, and this is a bit of a controversial topic, but just in brief, there are differing opinions, but many of us feel that a vaccine that could induce CD8 as well as CD4 cellular immunity as well as humoral immunity in an RSV naive population like, older infants and toddlers would be ideal. So thanks.
Great. Thank you, Dr. Karen. And we're going to have to end now. We're at time. I know there are important questions that came in from Robin Bielik, Laura Hammett, Kyra Mehta, but I apologize. We're going to have to wrap up now. And I really thank our speakers for today for your wonderful contributions, and for all of our listeners. I think there were up to 235 folks on the call. So thanks to all of you, and we will celebrate the great successes of vaccines during this World Immunization Week. So thanks to all of you.